This video is about Edexcel B1 Biology, Classification, Variation and Inheritance. This is one of 3 topics and therefore 3 videos that I'll make based around this unit of B1. So the first area starts with classification, moves on to variation and reasons for it, and inheritance and the ideas of evolution I sort of tied in before we get up to inheritance. So classification is the organisation of living things into groups. So you can think of organisms. We can think of like going into a supermarket and knowing that things are on certain aisles or certain areas like produce, or in the frozen aisles, or in dairy. So again, with animals we classify them using this to allow to see how they're related to each other. So organisms that are classified are based on how closely they're related. So you need to know the categories for classification. An easy way to remember them is King's Play, Chess Song, Fine Grain Sand, or Kevin Peterson, who many of you might not remember. Kevin Peterson's cricket often features giant sixes. And if you don't know who Kevin Peterson is a cricketer who was born in South Africa but played cricket for England and hit a lot of big sixes. So that allows you to figure out the order of kingdom, phylum, class, order, family, genus, species. So organisms within the same species are more closely related than organisms within the same genus. So if they're in the same species, they're close together in the same genus. So it starts off at peak over and spreads out, but if these are close together, if they're in the same species, they're close together than if they're in the same genus. So organisms in the same genus are therefore close together in the same family, and organisms in the same family are close together than those in the same order. So there are 5 kingdoms that we need to know about. So in terms of kingdoms across the top, there are 5 main ones that we need to know. We need to know the features of each of these, the main characteristics. So we've got Plantae, Animalia, fungi, Protista, and Prokaryotes. So Animalia, or animals, they have many cells. They generally don't have cell walls, don't need chlorophyll, and they feed heterotrophically. So I'll go through these words in terms of heterotrophically and autotrophically and saprophytically in a second. Plants, again multicellular, but this time they do have cell walls, they have chlorophyll, and they feed autotrophically. Fungi and multicellular have cell walls, they don't have chlorophyll, and they feed saprophytically. Protista, unicellular, so only one cell, have a nucleus, and prokaryotes unicellular have no nucleus. So these are key definitions that you need to know. Make sure you make a couple of notes on each of those. In terms of what these words mean, so heterotrophically means they cannot make their own food, so they eat other food essentially. Autotrophically means they produce their own food, and saprophytically means they feed off dead or decaying organisms, so they digest the food externally. Multicellular means they're made of just one cell, or more than one cell, sorry, and unicellular is made of just one cell. Right, so we've got some definitions that we've taken straight out of the specification. These are words that you do need to know. So they do crop up every now and again, normally as multiple choice questions, but it all comes from that classification order. So species are groups of organisms that have many features in common. Genuses contain several species with similar characteristics. Family are made of several genera, which is the plural of genus. Orders comprise of several families. Class comprises of several orders. Phylum comprises of several classes. And the 5 kingdoms, as we've just listed. Remember, most organisms are difficult to classify. That's because they have features from many different species sometimes. Most scientists agreed that viruses are non-living organisms. Non-living organisms cannot be classified. So if you ever ask anything about viruses and why they can't be classified, it's because they are non-living organisms. So 2 of within the phylum Chordatus is made of 2 main areas, and they are vertebrates and invertebrates. So we've got 2 main animals. So we've got those with a backbone and those without. So those with a backbone are called vertebrates, and those without a backbone are called invertebrates. So backbone and internal skeleton, invertebrates don't. Okay, vertebrates are divided into 5 groups called classes. Okay, so you've got mammals, reptiles, fish, amphibians, and birds. Mr. Fab. Okay, so they're the classes that make up vertebrates. Vertebrates are placed into placed in groups based on the following criteria. So like mammals can do certain things, and reptile can do certain things, fish can do certain things, amphibians can do certain things, and birds can do certain things. So to give you an idea of these, so I'm trying to find the mouse. There it is. They're based on how they absorb oxygen. so like gills or skin or lungs, how they reproduce, that's externally, internally, whether they lay eggs, whether they give birth to young and feed milk. Okay, that's what mammals do, remember. How they regulate the temperature, so homeotherms, they're warm-blooded, so homeostasis, what takes place there, and we do have a section on homeostasis in the next video. Or if they're cold-blooded, so in other words, their body temperature is the same as their surroundings. So picotherms, essentially, they use their surroundings, they go in and out of the shade or the sun to get warmer or colder. So the rules for classification are constantly being changed as new species are being discovered, and some of these don't fit into the categories that we automatically set up. A good example of this is something called duck-billed platypus. I've got a picture of one of these to the left over here. And it's difficult to classify as ducks and a bill like a duck and a tail like a beaver. That's difficult, but it's a mammal because it suckles its young, so in other words, it gives milk to its young, and there's homeothermic as well. However, it does lay eggs, so it kind of doesn't fit into any of those categories. So this is why organisms are difficult to classify. When we get onto species and breeding, we can start thinking about how sometimes, again, these areas are muddied. Sorry, I didn't mean to do that mark there. Let's get rid of that. So, organisms of the same species can interbreed, and they can interbreed to produce a fertile offspring. Not all organisms interbreed, and some organisms reproduce asexually, so they're still within the same species but with themselves. Fertile hybrids can occasionally be produced when offsprings from organisms from different species breed. So this happens often in ducks. So different species of ducks can produce a fertile offspring. So this contradicts the definition of species. However, the two species differ in many ways genetically. So in other words, they differ genetically. Members of the same species don't always look the same. Variation. So all of us are human, but we don't always look the same, because of the fact we have variation amongst our species. For example, every breed of dog looks different, however they are all the same species. A ring species is a group of a related population that live in neighboured areas. So these are often interbred and produce fertile offspring. So populations live far apart can't do this. So ring species are where you have related populations and they're close together. It's difficult to tell if the populations are from different species because of the fact they live so close together and they can produce fertile offspring. So that they are a very, very piece of clear evidence for evolution, ring species, because it happens very clearly there, because you can see how it's taking the best suited characteristics from both. Those that take the best suited characteristics from both are more likely to survive. Binomial system. So in the binomial system, species are given a two-part Latin name. The first part refers to the genus, the second part is species. So it's genus before species, just like in the classification order. So Homo sapiens, Homo is the genus, while sapiens is species. It allows scientists to clearly identify species and also study them, and therefore conserve them and target the conservation effort to make sure that we don't lose certain species. All of this has been allowed by using the binomial classification as a clear way of naming species. The keys are the way we actually identify animals. They consist of a series of questions that allow you to identify an organism, and after answering each question your options are limited. So a bit like Guess Who. Okay? Keep going until you get to one organism. So let's get on to variation. So variation is where organisms in the same species have a few differences. Different colour hair, eye colour, skin colour. And there's two main causes for variation. So we've got the genes, that's genetic variation, or the environmental variation. Genetics are due to mutations. Okay, so it's not necessarily anything we've inherited, but also it can be something we've inherited from the parents. So eye colour, blood group, or inherited disorders, they're all of those are genetic variations. Environmental variations are things we acquire, so things like scars or having a suntan. Well, both of these can cause variation. Most variations occur due to a change in the environment and a genetic factor, like height and weight. So we can categorize these two types of variation into two further categories. And these are called continuous and discontinuous variation. So continuous variation is where there's no distinct categories, so you can't plot a bar graph. Tend to be quite qualitative, so in other words using numbers, controlled by lots of genes, strongly influenced by the environment as well. Discontinuous variation has distinct categories. Okay, so like blood type, colour of a petal, something like that. Evolution. This is where Darwin took variation and really tried to understand why variation has come about and why certain species can really survive where they are. So some organisms have adapted to survive in extreme conditions like deep sea, volcanic vents, or the polar regions. Some deep sea fish emit light from parts of the body. This light attracts prey which they then feed on, and they have huge mouths that they can drag along the seabed to pick up loads of food that's there. But also they have huge eyes that allow them to see in the dark. They also have feelers which help them locate the prey, so they've got feelers that they sort of drag along the floor. Chemicals from volcanic vents support bacteria that are able to make their food using chemical energy, so that's chemosynthesis. The bacteria are at the bottom of the food chain, so they're producers right at the bottom, and the conditions are extremely hot and they're at high pressure, so normally these bacteria just die away, but they're better suited to survive. The bacteria living in the vents must be specially adapted, so they're adapted for those conditions, to survive in those conditions. Polar bears are rounded and have a small surface area to volume ratio. They have thick layer of blubber, acts as insulation and as an energy store, and they have these thick hairy coats. All of these prevent heat loss. They also have big feet to prevent them sinking into the snow or breaking the ice. They have white fur for camouflage, and penguins have a similar adaptation to polar bear, but they also huddle together to prevent heat loss. Streamlined body to reduce resistance, water resistance when they're swimming, and they have oily feathers also to prevent this heat loss. So these organisms are adapted to their environment, to survive in their environments. There will always be more organisms born that can survive, and those organisms that are best suited will do so and pass on their genes. This is known as survival of the fittest or natural selection. So we're getting into Darwinism now. So evolution is this idea that Darwin came up with, linking it to natural selection. That's what Darwin really did, linked it to the idea of natural selection. This is a slow process, remember, it's not quick, it's not sudden, it takes time. What Darwin said is that there's this process that we have to go through, it's VOSSAS, okay, are the letters that you need to remember, the 6 letters that till the start of this. So VOSSAS, sorry, SAG, sorry, I always forget that last bit. VOSSAG. So you've got variation, so populations of variation within them, overprediction. So some organisms produce more young that will actually survive to adulthood. Then you get this struggle for existence taking place because they're rapidly increasing in size and they're competing for food. So in other words, competition comes about. So survival. Those that are more likely to survive will survive. Advantageous characteristics inherited, so those that are more likely to survive pass on this advantageous characteristic they had, and then that leads to a gradual change over time. So if you imagine monkeys that had 5 fingers, and they were grabbing hold of trees, they'd keep slipping off because they don't have a thumb to push against the 4 fingers with. They would therefore fall off and die, and if there was a competition for food, they would be less likely to get it, compared to a monkey that had 4 fingers and a thumb. The monkey that had 4 fingers and a thumb would be more likely to survive, therefore would be more likely to reproduce, pass on that advantageous characteristic, and therefore would be inherited, and gradually over time all the monkeys would end up with 4 fingers and a thumb. So there's evidence for evolution. The main piece of evidence is DNA research, or one of the main pieces of evidence is DNA research. Over time, organisms diverge away from each other in terms of DNA. This means that any organism that have recently dived away from each other, so chimps and humans, should have similar DNA. And guess what they do? Resistant organisms, okay? So an example is poison morphine, was used to kill rats. Certain genes in the rats give them a resistance to this. These rats are more likely to survive, and therefore they breed. Now there's entire populations of rats that are resistant to warfarin. Same thing happens with MRSA. So MRSA was used. It says there's a whole academic within hospitals, and they use certain antibiotics to try to get over that. But what actually happened is the MRSA became resistant to the antibiotics because some of them survived and then reproduced. So they actually started to use antiseptics instead to completely kill all bacteria before it actually started getting into the areas where MRSA could come about. So again, the idea of MRSA led to this idea of, so it helps us understand evolution in action. So whenever we come across these new ideas, there's ways we're going to verify this, these ideas. And the scientific community does this in three main ways. They have scientific journals, they're like magazines that scientists write, and they sort of share their ideas. And then other people can sort of test their experiments to see whether they can do them. There's the peer review process, where scientists check that other people's experiments are valid and carry them out to the highest standard. And there's scientific conferences where people present their findings to other scientists and they discuss. So, moving on from evolution, we have speciation. So speciation is where evolution leads to new species. This occurs when the populations of the same species are so different they can no longer breed together to produce fertile offspring. So like you've got two species, A and B, and they reproduce and they produce C, and C is another species. Now A and B become so different from each other now that they no longer can actually reproduce to produce a fertile offspring. So this has happened because those two organisms or species have sort of been separated over time by like an earthquake or a flood or a volcano. So over time, the conditions on either side of this barrier will be a bit different, and therefore they won't be able to produce anymore. So eventually the populations will have so many differences they can't breed with each other to produce fertile offspring. So now you end up with two different species. So you have species A, it splits into two species A and species B. Then they no longer can actually, their characteristics go off in different directions, and they no longer can reproduce. Think of it like Pangea. So Pangea is what the Earth was actually like with the continents all in one place. So you had all the animals that would go on to become the Australian animals there, you had all the African animals there, and then Australia came off, and there was a big separation, and they evolved differently, and speciation took place. You had different species in Australia to those in Africa, even though if you go back they are related in some ways, but if you try to interbreed the two, it doesn't work. So let's start going inside the nucleus. Inside the nucleus you have chromosomes. We have 23 pairs of chromosomes, that's 46 altogether. Chromosomes tell us essentially our different characteristics: hair colour, eye colour. That's pretty much, all our inherited characteristics. That's the code for it in the chromosomes. So chromosomes are made up of genes, and each gene essentially defines that characteristic. Okay. So these are different versions, and they're called alleles. So you have one version that leads to brown hair, and one version that leads to blonde hair. So different versions of the same gene. So one gene talks about hair color, but one allele of that is brown hair, and the other allele is blonde hair. Each chromosome within a pair will have its own allele. So you might have two of the same allele, you might have two different alleles, and we'll get onto that in a bit when we talk about inheritance. Chromosomes are a long length of DNA essentially called up. So each section of DNA is called a gene. The DNA molecule has this double helix shape here. And you learn about how we came up with that structure in B2. Right, so to understand genetic diagrams you need to understand a few key words. There's a dominant allele, so that's the one with the characteristic that is shown. So if you've got brown hair, the dominant allele is brown hair. The recessive allele, so that's the one that's not shown. So if you had brown hair and blonde hair alleles, brown hair was shown, so that's dominant allele. Blonde hair wasn't shown, so that's a recessive allele. Homozygous. Homozygous is when two alleles for a particular gene are the same. So if you have brown hair, that's what that means. Heterozygous, so when the two alleles for a particular gene are different, so that's like brown hair, blonde hair. Phenotype is a characteristic you have, i.e. brown hair. Genotype is the alleles that you have. So big B, little b, so brown hair, blonde hair. Zygote is a fertilized egg. Gametes, or sex cells, have only one type of allele, but all of the cells have two. The dominant and recessive alleles are presented in your genotype, so the alleles that you have. The dominant will always show its characteristics when we go to phenotype. Right, let's get into some ideas of inheritance then. So over here we've got Punnett squares, and we've got inheritance diagrams here as well. These can be used to show how characteristics can be inherited. Both diagrams clearly show specific combinations of alleles which can be produced. So we've got the idea of black body and grey body. So we've got big B, little b, little b. So these two join together to make a big B, little b. Because the big B is dominant, you end up getting the black body. That there is going to be big B, little b, again black body. Big B, little b, black body, big B, little b, black body, because the B is dominant there. Okay, so you can see that all here, all of them have the same big B, little b. 100% chance of black bodies, 0% chance of grey bodies. If I have a black body over here though, that's big B, little b, and a black body over here, and that's big B, little b as well, there's a percentage chance that these could end up with a grey-haired fly. So, big B, black body, big B, little b, black body, carries it on, it carries that to the next generation. Big B, little b, again, carrier, carries that on to the next generation. Little b, grey body. So because these are both carriers, you could end up with a grey body fly. I'd just like to point out that in the first diagram, again, these are 100% carriers. 100% black body, but 100% carriers with that grey body. So cystic fibrosis is a genetic disorder. So you can do it in a Punnett square like above, or you can do it in these cross diagrams. And it's the result of it is it produces a large amount of mucus. that blocks sort of air passages in the gut and the pancreas. Okay, so it means that people with cystic fibrosis find it difficult to breathe. They also find it difficult to digest food. Okay, so you need to take medication for that, and also take massage for it as well. It's produced by a recessive allele. So the diagram on the right shows a genetic cross for 2 carriers of cystic fibrosis. Okay, the ratio for sufferers to non-sufferers is 1:3. Okay. So normal, don't really like that word, but that's what it's saying. So remember, it's phenotype, that's a genotype. Both of these are heterozygous. Okay, remember, both of these are heterozygous. And if I go a bit more, that's homozygous. That's homozygous. Both of them are homozygous because they both have the same, but they produce heterozygous offspring. So right, let's go with this one. So big C, little c, big C. So little c, big C. This one over here is big C, little c, and this one over here is little c, little c. So these are carriers because they have the recessive allele, but this one over here is a sufferer, but both recessive. He ends up having cystic fibrosis. Sickle cell anemia is also caused by recessive alleles. Okay, so again, they can need smallest for it to happen. So you can see the crossover here, as in one of them ended with smallest. So 3:1 for sufferers, 1 sufferer for 3 non-sufferers. However, you do have 2 carriers, 50% chance of being a carrier, but only because they're both carriers. It's worth spending some time to make sure you understand those. And the last little section is pedigree analysis. So these diagrams are always quite tricky, you need to take your time. So pedigree analysis is like a family tree. Let me zoom in on this picture again. There we go. And it helps you understand inheritance. So in this situation over here, U, V, and Y are all children in the second generation. So the first generation parents of U, V, and Y are all children. X is a child of U and Y. Sorry, U, V, and W are all children. So X is a child of U and so X is a child of U and Y is a child of W. Sorry, I should say X is a child of U and Y is a child of W. From this tree, you can tell that the allele is not dominant because there are carriers of the allele who are not sufferers. Okay, so it cannot be dominant. Okay, it must be recessive. So you've got carriers, but they're not sufferers. Okay, so you can see that you've got carriers, but they're not suffering from it. They're not affected. Therefore, they it must be a dominant allele that's causing this. So in other words, there's a 0% chance of a child of V and her husband being a carrier or a sufferer because neither of them is a carrier or a sufferer. They're normal. They have no carriers, no sufferers. It's a 50% chance that you, that a child of you will be a carrier, okay, because of the diagrams you did above. And there's a 25% chance of being a sufferer, just like I said before, a 1:3 ratio or 1 in 4 chance. There's a 100% chance that a child of W will be a carrier because of the fact that W is a carrier and they're having a child with someone who's affected. Okay, and that's the end of topic 1 of B1 GCSE Edexcel Biology.